When a patient suffers massive hemorrhage, the body rapidly loses intravascular fluid. As venous return drops, cardiac output collapses. Without enough volume, the circulatory system fails to perfuse tissues with oxygen, driving the body into hypovolemic shock. To reverse this crisis, pharmacology relies on two distinct interventions. This schematic illustrates these two options: biological human blood products on the left and synthetic plasma substitutes on the right. Blood products are active biological therapies. They are administered to restore the living cellular components a patient has lost, such as red blood cells, platelets, and coagulation proteins. Plasma substitutes, conversely, are biologically inert. They rely on purely mechanical fluid dynamics to expand the intravascular space. This distinction forms the core rule of resuscitation fluid mechanics. Blood products replace what is missing, while plasma substitutes mainly replace the volume. To understand how to deploy these interventions, we will first examine the biochemistry required to keep human blood alive outside the body, and then dissect the physics driving synthetic volume expanders. Modern resuscitation requires a mastery of both biological tissue preservation and synthetic fluid dynamics. Whole blood is a specialized fluid connective tissue consisting of cellular components suspended in liquid plasma. If you remove the clotting factors from that plasma, the remaining liquid is called serum. Because whole blood contains living cells, it must be extracted in a highly controlled, sterile environment, following rigorous physiological screening of the donor. Collected units are immediately typed according to the ABO and Rh systems. Administering incompatible blood causes the recipient's immune system to attack the donated cells, resulting in a fatal hemolytic transfusion reaction. The fluid also undergoes mandatory screening protocols to detect transfusion-transmissible infections like HIV, Hepatitis B, Hepatitis C, and Syphilis. Once collected and screened, the blood faces a severe biochemical challenge, preventing the fluid from coagulating and the cells from degrading. This is managed by adding a preservative solution known as CPDA-1. The C stands for citrate. It actively binds to ionized calcium, physically trapping it to halt the coagulation cascade and keep blood fluid. Next is dextrose. It provides the direct energy substrate needed for red blood cell glycolysis to generate ATP. The P is for phosphate. It buffers the chemical environment, maintaining precise pH levels. Finally, adenine. It supports continuous ATP synthesis, drastically extending red blood cell survival during prolonged storage. Collecting blood is an exercise in biochemical engineering. We carefully manipulate its chemical environment to suspend tissue degradation and keep the cells viable outside the human body. Pharmacopeal guidelines require whole blood to be stored under strict, continuous refrigeration, typically between 2 and 6 degrees Celsius. This temperature range serves a specific physiological purpose. Cold environments decelerate cellular metabolism, reduce the rate of glucose consumption, and severely inhibit the growth of contaminating bacteria. The temperature must never drop below freezing. Ice crystals will form inside the bag and physically perforate the red blood cell membranes, destroying the tissue. Conversely, if the temperature rises too high, rapid metabolic activity causes ATP depletion, membrane damage, and ultimately hemolysis, the rupturing of the red blood cells. Even under perfect storage conditions, the blood undergoes gradual biochemical degradation, known as storage lesions. This includes reduced pH, increased plasma potassium, and increased red blood cell fragility. To mitigate these storage lesions and maximize the utility of every donation, modern blood banks use centrifugation to separate whole blood into distinct layers based on density. This yields specific clinical products: packed red blood cells to restore oxygen capacity, concentrated platelets to fix clotting deficiencies, and Cryoprecipitate, a plasma derivative enriched with factor VIII and fibrinogen. By fractionating whole blood, physicians avoid treating a specific deficit with a blunt instrument. Patients receive exactly the cellular components their condition requires. The plasma fraction isolated from that whole blood is approximately 90% water. The remaining 6 to 8% consists of critical coagulation proteins like albumin and fibrinogen. This liquid plasma can be processed through lyophilization.
As seen here, the plasma is frozen solid and subjected to a vacuum, causing the ice to undergo sublimation, turning directly into vapor. The removal of water leaves behind a stable, dehydrated powder containing all vital proteins. This formulation solves massive logistical hurdles. It is exceptionally light and features a vast storage lifespan without any requirement for continuous refrigeration. For decades, this made dried plasma a vital asset in military and austere emergency medicine, where maintaining a refrigerated cold chain is physically impossible. By extracting the water, scientists preserve the biological scaffolding of the plasma. As long as sterile fluid is available for reconstitution on the battlefield, the proteins can be reactivated on demand. When human blood products are unavailable, physicians must turn to mechanical interventions to keep the cardiovascular system online. These are plasma substitutes. They are strictly volume expanders. They do not contain red blood cells, they do not contain platelets, and they provide zero immune or clotting functions. They operate using colloid osmotic pressure. In this cross-section, large synthetic colloids are injected into the depleted bloodstream. These colloid molecules are physically too massive to pass through the semi-permeable membranes of the blood vessel walls, remaining trapped inside. Because they are trapped, their concentration creates an osmotic gradient. They recruit and retain water molecules, pulling them from surrounding tissues into the bloodstream. As water floods in, the plasma volume expands, temporarily reversing the mechanical parameters of shock. These substitutes are grouped into two primary categories, crystalloids and colloids. Crystalloids, like normal saline or Ringer's lactate, contain very small ions. They expand volume briefly, but those small molecules rapidly leak out of the vascular space and distribute across all body fluid compartments. Colloids, conversely, contain the large, heavy polymers required to exert strong, sustained oncotic pressure and keep the fluid precisely where it is needed, inside the veins. Plasma substitutes do not heal the tissue deficit. They leverage the basic laws of osmosis to prop up a failing circulatory system when real, biological fluid is out of reach. There are 4 major colloids utilized in medicine, and choosing between them requires calculating the specific risks of the underlying polymers. The first is dextran. These are high molecular weight glucose polymers produced by microbial fermentation. They are highly effective at oncotic volume expansion. However, dextrans carry severe clinical limitations, frequently triggering anaphylactoid hypersensitivity reactions and actively interfering with natural coagulation. Next are gelatin preparations. Gelatins provide rapid volume expansion, but for a relatively short duration, and they still carry the risk of severe hypersensitivity. The third synthetic option is hydroxyethyl starch, or HES. HES is a powerful modified starch polymer, but its use in critical care is now heavily restricted globally because it poses severe risks of acute kidney injury and coagulopathy in vulnerable patients. These 3 synthetic options stand in sharp contrast to the fourth colloid, human albumin. Albumin is the exception. Because it is a natural human plasma protein derived through fractionation, it safely exerts colloid osmotic pressure while also performing vital biological binding and transport functions within the blood. Synthetic colloids provide a brutal mechanical push to keep blood flowing, but they cannot replicate the complex biological safety of natural human proteins. This matrix synthesizes the pharmacological divide. Looking at their source, blood products are human-derived, whereas substitutes are synthetic or purified proteins. Red blood cells, platelets, and clotting factors are present in blood products, but strictly absent in substitutes, and their primary clinical purposes sit on opposite ends of the spectrum. One replaces specific missing components, while the other merely expands circulating volume. From the donor's arm to the emergency IV, modern resuscitation hinges on exact science. Chemical preservation and fractionation keep complex cellular tissue alive, while lyophilization and heavy coloners offer a mechanical backup when cold chain logistics fail. Blood products replace what is missing. Plasma substitutes mainly replace the volume.